again. برحب بكم مرة تانية. We are back. So let me share the screen with you again. So here we will just go back to the cellular currency exchange. What were we at last? We just stopped at the 4 requirements of respiration, which are a TCA cycle or a Krebs cycle, electron transport system, coupled with an outside electron acceptor and a transmembranous ATPase enzyme. So there are 4 conditions for respiration, not including molecular oxygen. Molecular oxygen is just one option for the cell. If you remember well. So what happens when you have the whole system, whether you have oxygen or you have anaerobic respiration with nitrogen, sulfur, and so on, then you go to the electron transport chain. You want to exchange electrons and get energy. So you have saved a lot of electrons during this process. There has been a lot of electrons carried on NAD or sometimes on FADH. NADP in some of the biochemical pathways. You will never go through it this year. هتسمعوا عنها السنة الجاية في biochemistry. So now we want to know how to exchange this reducing power, or the reducing power is always electrons, of course, and how to get it into the form of ATP that you can use. And this is what we call the oxidative phosphorylation, where the final it happens after all respiration is over, and you get a lot of energy from this. Usually aerobic gets a little bit more, but also anaerobic, both aerobic and anaerobic respiration generate much more energy than fermentation, because fermentation only uses اللي هي الgenerator that we called substrate level phosphorylation. So let's go through this really slowly. So first of all, you have 2 currencies. You have electron currency, which is the electron carriers, NAD والحاجات اللي زي كده, and you have an energy currency, which is the ATP. So you want to do this exchange, and most of the energy producing reactions, as you will know, go through these intermediates اللي هم ال NAD, NADPH, and the flavoproteins. So near the end, everything in the bacteria is happening in the cytoplasm. In humans and eukaryotes, there is a lot of compartments, there is mitochondria, but in bacteria, it's really simple. And this is what we mentioned in the cell structure lecture. لو تفتكروا المحاضرة الثالثة بتاعت ال bacterial cell structure, قلنا إن cell membrane is the energy generation location, or that's where the energy is formed, or ATP is formed. So we have this complex system, amazing system of cell membrane proteins. There is a lot of elements of this electron transport chain, and the best thing is to really watch it in a video. So let's see this in a narrated video, and I will comment on it after.
During glycolysis and the tricarboxylic acid cycle, oxidation of organic molecules results in production of reduced coenzymes such as NADH.
تمام، يبقى احنا كده اول خطوه بنقول ان coenzymes دول اللي هم اللي قلنا عليهم في الاول، او carriers اللي هم بيساعدوا الانزيمز بتتحمل الالكترونز على NADH وما شابهه.
These coenzymes transfer hydrogens to the electron transport chain, which is located in the bacterial cell membrane. A hydrogen consists of a proton and an electron.
طبعا الحته دي نركز فيها جدا. الهيدروجين احنا كلنا عارفين يعني طول عمرنا it's just a proton and an electron. والالكترون ده هو اللي هيلعب بقى طول الالكترون ترانسبورت تشين دي. ليه؟ لان انت كل ما بتعمل اكسبورت لالكترونز البروتون بيبقى لوحده. The proton is left alone, so it will be kicked out of the membrane. So you will have a lot of protons out of the membrane, and all the electrons are moving through the electron transport chain. Then all of a sudden, all these protons, I want you to watch that, will come back inside the cell and then they will be binding to oxygen to form water, but they will generate through this amazing instrument, the ATPase pump. Let's see.
The electron transport chain consists of a series of special electron carrier proteins that shuttle electrons from NADH to a terminal electron acceptor, such as oxygen. Electrons enter the electron transport chain when NADH transfers its protons plus electrons to a membrane-embedded carrier protein.
What happened here? So here is NADH, which is loaded with electrons. It gives away its electrons here to the ETS, the electron transport channel, and the electron will start going down the channel. And every time a proton is released, because the NADH has a proton and an electron, this is how it carries the electron. It will become NAD.
The electrons are sequentially carried along the electron transport chain.
Here we go again. Just watch this again and again. I can get the movie a little bit back because it's really interesting. All these protons are kicked out, and there will be a majority of. That's why they call it chemiosmosis. Remember, osmosis means there is a differential concentration. كأني بطرد كل البروتونز بره، بعمل يعني positive charge عالية من البروتونز، and the protons لما يعملوا كده هيرجعوا تاني، وده اللي كنت بسميه السد العالي. They will come here and go down the ATPase، وهنشوف ده هيحصل ازاي. Let me back off just a little here. نشوف ده تاني ومش هتكلم تاني.
Electrons to a membrane-embedded carrier protein. The electrons are sequentially carried along the electron transport chain, while the protons are shuttled to the outside of the membrane.
Electrons down the chain, protons out of the membrane.
Some of the electron carriers, such as coenzyme Q,
that's one example of the carriers, the electron carriers. It's called coenzyme Q.
Enzyme Q accept a proton from the inside of the cell membrane.
See, it takes the proton here.
As it accepts electrons. The proton is then transported through the membrane as electrons move down the chain. This increases the proton gradient across the membrane and enhances the proton motive force.
So this just shows you the details, but we don't have to know this by detail, just the videos. And you have all the links to the videos. You can watch them later on YouTube.
During aerobic respiration.
And here we go. أول بروتون بير دخلوا أهم، اتشتين مع إلكترونين مع أكسجين. الأحمر ده أكسجين. This is O2. O2. O and O. And then they will make water.
The last carrier protein transfers a pair of electrons to oxygen at the end of the electron transport chain, and water is formed. The enzyme ATP synthase utilizes the energy of the proton motive force to synthesize ATP.
And here we go. This is how ATP is made. All right.
During glycolysis and the tricarbox.
So here is what happened again. In very simple terms, there is equal gradient. This is what we call the proton motive force, PMF. That's what we just watched in the video. There is equal protons inside and outside of the cells. Okay, so here the ATP can be broken into ADP and phosphate. We don't want that. We want the protons to be out more. So when you have a lot of protons out, this is reversed. Again, when there is equal protons inside as an outside, the ATP gets normally degraded because the cell needs it. It just uses it. However, when you want to generate more ATP, you use the electron transport chain, as you just saw, to kick protons out, and then protons will come back and will regenerate ATP through this pump. We can see this one more time. Here the video is from mitochondria. So it's not prokaryotic, but it still shows you how the gradients are formed. Very interesting.
Concentration gradients are a key component of the biological world. The potential energy from these gradients is often used to perform biological work. Here we will focus on hydrogen ion concentration gradients. Hydrogen ions are also known as protons. A gradient exists when there is a higher concentration of a molecule in one compartment compared to a neighboring compartment. This animation will demonstrate how the potential energy that results from a hydrogen ion gradient uses ADP and inorganic phosphate, also known as PI, to synthesize ATP. This process involves an enzyme complex called ATP synthase. Gradients and the potential energy they create are key aspects of the biological world. A good example of the use of a gradient occurs in the mitochondria when ATP is synthesized. ATP is...
Here it talks about mitochondria because it's just an example, but it's the same exact pump. So it's really good. Sorry. It's really good to watch it. I'm sorry, the video was skipped.
In this nated subunit.
آسف جدا على اللي حصل.
The large difference in the number of hydrogen called ATP synthase. Gradients and the potential energy they create are key aspects of the biological world. A good example of the use of a gradient occurs in the mitochondria when ATP is synthesized. ATP is synthesized by ATP synthase, a large complex of membrane-bound protein. Here we see ATP synthase, along with other membrane-bound proteins.
طبعا الموليكيول بتاعت ال ATP synthase دي من معجزات البيولوجي، يعني اكتشافها اساسا كان حاجه عظيمه جدا انهم اكتشفوا قد ايه هي يعني it's a very sophisticated machine inside our body. It looks like really a motor and a dynamo in the same time. It generates ATP and it breaks down ATP too.
It's a large difference in the number of hydrogen ions on the two sides of the membrane. This difference is a hydrogen ion or proton.
The proton motive force when there is more protons outside.
Concentration gradient. The energy associated with this gradient is used to synthesize ATP from ADP and PI. This occurs at the ATP synthase complex. One hydrogen ion enters the
there is always a problem at this stage. I'm sorry. Let's see.
Hydrogen ions have crossed the membrane. Another molecule of ATP will be made. In this example, the hydrogen ion gradient is large enough to produce 6 ATP molecules. Please watch as the remaining ATP molecules are synthesized.
ركزوا جدا على البروتون وهو بيدخل. الحاجات البينك اللي بره دي بروتونز. كل ما بيدخل بروتون والاي دي بي اهو جاي with 2 phosphates it becomes ATP. See?
The process has now completed, and the result is an equal number of protons on each side of the inner membrane. Without a gradient, there is no more energy available to make ATP. In biological systems, however, a gradient is always maintained. The mitochondrial hydrogen ion gradient is generated as electrons pass through 3 membrane complexes. That process can be seen in the mitochondrial electron transport chain animation.
So the mitochondria, if you remember, it's exactly like a bacterium. So it does the electron transport chain in its membrane. Let's see more about the ATP synthase pump.
ATP synthase is a molecular machine that works like a turbine to convert the energy stored in a proton gradient into chemical energy stored in the bond energy of ATP.
You can see here the ATP pump. Again,
the flow of protons down their electrochemical gradient drives a rotor that lies in the membrane. It is thought that protons flow through an entry open to one side of the membrane and bind to rotor subunits. Only protonated subunits can then rotate into the membrane.
هي البامب دي الموتور العظيم ده اللي عندنا في السيل ممبرين ما بيتحركش غير لما البروتون بيدخل ويزقه بالطاقه الالكتريك بتاعته.
From the static channel assembly. Once the protonated subunits have completed an almost full circle and have returned to the static subunits, an exit channel allows them to leave to the other side of the membrane. In this way, the energy stored in the proton gradient is converted into mechanical rotational energy. The rotational energy is transmitted via a shaft attached to the rotor that penetrates deep into the center of the characteristic lollipop head, the F1 ATPase.
حاجة معجزة فعلا يعني. دي هنا نموذج زي من الصلصال كده. It's a model of this pump, but it shows you really in detail. And scientists have calculated all this in very much detail. They crystallized it. They watched the proteins under X-ray diffraction and figured out the structure. So it was really one of the biggest achievements of biology.
Which catalyzes the formation of ATP.
Of course أنا مش عايزكم خالص تحفظوا كل التفاصيل دي، بس استمتعوا بيها يعني. حاجة فعلا رائعة. أنا عارف يعني حاسين إن أنا ببالغ، بس هي فعلا من الحاجات الجميلة جدا في الخلية. نبص تاني كده.
Formation of ATP.
بصوا ادي ال ATP بيتكون هنا زي ما شفناه في
The F1 ATPase portion of ATP synthase has been crystallized.
زي ما قلت لحضراتكم it was crystallized. عرفوا بالظبط العمود الفقري بتاعها وعرفوا كل حاجة عنها. جميل جدا.
ATP synthase is a molecular machine.
فكل الفيديوهات دي, all these videos you can find on YouTube. In addition, there is an extra one with music. It's like an ATP song. ممكن تتفرجوا عليها. All right, let's summarize now. So, heterotrophic catabolism, one more time. Catabolism is the breakdown of larger molecules into smaller molecules to generate energy in the form of ATP. And other than the small molecule and energy, you get a lot of electrons that you transform in the end with the electron transport system or electron transport chain through kicking protons out. يعني عن طريق electron transport chain دي بتطرد البروتونز out بره الخلية so that you can generate a lot of ATP. فالoverall بقى product بتاع aerobic respiration in the glucose will be oxidized. This is a very يعني a summary of so many steps. Glucose is oxidized. As we saw in glycolysis, glucose بيبقى pyruvate, وبعدين pyruvate تخش في PCA cycle. The outcome, the final outcome is 6 carbons in glucose become 6 CO2. Complete burning of the glucose, complete oxidization. Every carbon is oxidized now into CO2. Then there is a lot of electron carriers. 10 NADH, كل واحدة منهم will be used to generate ATP through the ETS. Also 2 FADH2 and 4 ATP. These 4 ATP are very important. How are they formed? Substrate level phosphorylation. ليه؟ لأنهم طلعوا they are early ATP. They are generated during the process of catabolism, not at the end. What about these 12 molecules, 10 NADH and 2 FADH2? They will generate more ATP. تمام? So 2 NADH from glycolysis. كل التفاصيل دي you don't need to know all these details. In eukaryotes, there is 38 ATP generated per 1 glucose molecule. In E. coli, it doesn't happen very efficiently. There is 26 ATP molecules per 1 glucose molecule for some details that you don't need to know. But bottom line, the aerobic respiration can generate a large amount of ATP. 26 is a lot, 26, 38. Anaerobic respiration, similarly, exactly the same. You have everything like aerobic except oxygen. So it uses electron acceptors other than oxygen, like nitrate, sulfate, or sometimes carbon dioxide that becomes methane. So some bacteria are anaerobic. They use carbon dioxide and generate biogas اللي هو الميثane. So all we need to know about anaerobic respiration for now is that it generates energy, but by not by putting the electrons on oxygen, إنما by putting the electrons on other inorganic molecules like nitrate, nitrite, sulfate, and so on. نيجي بقى للfermentation. Fermentation you know about since the second lecture, because you looked into an assignment and saw that bacteria makes a lot of good fermentation products like yogurt, alcohol, all types of cheese, propionic acid, acetic acid الخل. What is fermentation? Fermentation is a shortcut. يعني إيه بقى? The bacteria cannot or doesn't want to make a full respiration cycle. They just need a little energy. So they just get the energy only on the substrate level by partial oxidation. and then they start putting the electrons on an organic compound, not on oxygen or on CO2 لا أو NO2 لا، بيحط الالكترونز بتاعته على طول على organic compound ويخلص. فالorganic compound ده gets reduced أو يحصل له أي حاجة, and it gets transformed and give us everything that we see يعني from any fermentation product, cheese, drinks, and so on. So تاني, glucose enters glycolysis as usual and stops at pyruvate. Pyruvate is always there because it is, أيوة حد يقول كده معايا مع الفيديو, it's the universal intermediate. The universal metabolic intermediate is pyruvate. Whether you're going respiration or fermentation, there is always pyruvate. بعد كده بقى البيروفيت ممكن يتحول للاكتيت مثلا. تمام, ياخد electrons من NADH يبقى هو الالكترون acceptor ويتحول للاكتيت, يبقى the product always is reduced. حتى لو acid بيتحول لأسيد, الأسيد التاني بيبقى has some extra يعني a reduction of its parts. يعني لو هو acid عنده 2 COO groups مثلا, بيتحول لأسيد عنده COO group واحد. تمام, يبقى نقول تاني, احنا عندنا من الأول من ساعة ما ابتدينا البروسيس قلنا الجلوكوز goes all the way to pyruvate. It can continue to respiration through Krebs cycle, CCA cycle, or it can just go to a shortcut and the pyruvate will become a fermentation end products. What are examples of these fermentation products? Maybe this slide is not very clear, but you can find in your PDF quite clear and in the book. So there are 5 examples here. You don't have to memorize the 5. You can memorize 2 or 3 maximum. These are different types of bacteria and how they ferment. For example, there is Swiss cheese that you see in Tom and Jerry movies. بتشوفوا دايما Swiss cheese. Swiss cheese is characterized بإيه؟ جبنة سويسري. It always has these bubbles. These bubbles are carbon dioxide, and the cheese itself is an lactic acid or propionic acid. يعني, so propionibacterium is one organism that converts milk to lactic acid and then further to propionic acid and CO2, and a lot of CO2 is generated, so it causes all these bubbles in the cheese. Another type of organisms بقى is Aspergillus, the fungus, Lactobacillus, Streptococcus, أو Lactococcus. يعني 3 different types of bacteria that produce lactic acid and not much gas. فدول بيعملوا حاجات زي الشيدر cheese, اليogurt, even soya sauce is made by these bacteria. Saccharomyces is the most famous fermenting organism. مشهور جدا بالتخمير, عشان كده سموها الخميرة. والخميرة سميت كذا لأنها يعني بتصنع الخمر, والخمر يخمر العقل. هو ده الاسم العربي ليه. Saccharomyces cerevisiae. It generates different types of alcohol and carbon dioxide. That's why a lot of alcoholic beverages too may have bubbles because of the carbon dioxide that comes out, like wine and beer and so on. في بقى bacteria اسمها Clostridium. It ferments to acetone and isopropanol. So we can use bacteria to make nail polish removers وحاجات زي كده. Isopropanol is a very good disinfectant. So if you don't have enough ethanol for sanitizing, you can use isopropanol. Many of the sanitizers are made of isopropanol. And why not from ethanol? They try, يعني many people actually they're alcoholic. في ناس بتحب الخمور ومدمنة خمور, فبيجيبوا الإيثانول ده بتاع التطهير 70% ويخففوه يشربوه. That's why many of the sanitizers or antiseptics are made of isopropanol, so that people don't drink them. Finally, last but not least, there is Escherichia. Escherichia coli, طبعا, you heard about it, and Acetobacter, another type of bacteria. They can make acetic acid. All this from one molecule, glucose. So glucose can give you many other products depending on the bacteria that make it. So by now, this is the most important part of the metabolism lecture, and very important part, Nigma, highly important part of, it's the 3rd most important part of microbiology after the cell wall structure and after the growth. This is the comparison between respiration, aerobic and anaerobic, and fermentation. So we already went through it, but I'll just remind you. Which one requires oxygen? Only aerobic respiration. So bacteria that do aerobic respiration are usually obligate aerobes. Some bacteria are able to do both aerobic respiration and fermentation, and they're called facultative anaerobes. Anaerobic respiration is another option for bacteria. They burn glucose completely, but using substrates that are not oxygen, so they don't require oxygen. What type of phosphorylation here? There is substrate level phosphorylation in all of them. All types of metabolism, of catabolism, use substrate level phosphorylation, which means they generate a little bit of ATP in the middle of the reaction. كل الأنواع بتعمل substrate level phosphorylation بيطلعوا ATP في النص. But only respiration, عشان هي حتى اسمها respiration يعني oxidation. They use oxidative phosphorylation. What does it mean? Oxidative phosphorylation means that they generate energy in the end through يعني conversion of electrons to ATP. طبعا final electron acceptor هنا oxygen. هنا قلنا بقى كذا حاجة، نيتريت سلفيت سلفيت، كل مرة نقول لكم حاجة جديدة. هما كفاية قوي نيتريت سلفيت خلاص. بينما في الفرمنتيشن very important تحفظوا الجدول ده، بيجي كتير جدا في امتحانات. This table is يعني is very often a topic of the exam, whether as a complete the table or يعني you get the table with some spaces, blank spaces, and you have to complete them, or just through different questions like true and false, or just a question of compare fermentation to anaerobic respiration أو أي حاجة زي كده. So fermentation produces very little energy because it uses the electrons on endogenous organic molecule as electron acceptor. So they don't convert electrons into energy. They convert electrons, or they use electrons to reduce a chemical زي ما بيطلعوا أسيتون مثلا أو lactate أو أي حاجة. And there is only substrate level phosphorylation, so there is not enough energy. Anaerobic respiration can generate up to 36 ATP. Aerobic respiration generates from 36 to 38 ATP molecules. With some exceptions, مش مهم، خلينا في القاعدة. All right. Now, lithotropic catabolism, just one word. Lithotrophy means inorganic compound في الآخر زي nitrate, nitrite, and I don't want you to memorize this at all this year. Enough for you. So it just know that some bacteria, and they are usually aerobic, they just use inorganic compounds as electron source instead of getting their electrons from organic compounds. All right, so now we have gone through the entire chapter, and I'll just give you a very quick review, which we already solved if you were there last week when we met online. I solved these questions, but we can go through them quickly. They were unsocrative. So if you have a tube and you have bacteria growing in this tube, but the bacteria is in a room full of nitrogen, no oxygen, and then you started putting a tube here, a small tubing, and an oxygen source. زي ما كأنك كده غواص. It's like diving. Somebody diving, you have O2 here. So, and the bacteria started growing near the bottom. Oh. So what kind of bacteria are these? Obligate aerobe, obligate anaerobe, facultative anaerobe, microaerophile. Normally, aerobes will grow here. Anaerobes will grow here. But in this case, the room is full of nitrogen, so there is no oxygen. So whatever grows near the oxygen is definitely an obligate aerobe. ماشي? A bacterium that uses light as a source of energy, it's a photo something. كلهم photo. طيب ما استفدناش حاجة. They're all photo. I need more information. And glucose as a source of carbon. Is it phototroph, photoglucose, photo ما اعرفش ايه. طبعا there is no such thing as photoglucose. دي كلمة من تأليفي. Doesn't exist. So is it photoautotroph? No, autotroph means carbon dioxide. Is it photoorganotroph, heterotroph, lithotroph? Organotroph, it uses glucose but not as a source of carbon, as a source of electrons. Lithotroph طبعا بتستعمل inorganic فمش glucose. So we are left with photoheterotroph. تمام. The growth rate versus switch curve is symmetric. طبعا دي خدناها. This is chapter 5. There are two curves, one for pH. يعني there is nothing هنا. It's a piece of information and it was in the lecture. And there is temperature. Temperature, the curve is asymmetric because the bacteria die immediately when the temperature is a little bit above their optimum عشان طبعا بيحصل ايه protein denaturation, cytoplasmic coagulation. بينما ال pH is a symmetric curve, so the answer is B. A bacterium that has flagella everywhere around it is a, this one I will let you answer it because you already know it. It's in lecture or chapter 3. The following can grow inside the human body. This is a good question. We need to think about. مين من دول, which of these can grow in the human body? Is it hyperthermophiles? They need 90 degrees to grow. Do we have 90 degrees in our body? Of course not. So this is wrong. Acidophils, is there a place in our body where acidophils can grow? Yes, the stomach. Mesophils, what does it mean, mesophils? They need 37 degree temperature. What's our temperature? 37. So the answer is also, this is right too. So the answer is B and C. It's not all the above, and it is E. And I gave you a competition. It's too late to tell you the competition now. It's already in your classroom. So I hope some of you have solved it. There is a question on chapter 7 and a question on the prior chapter. And we can solve them together when we meet on any Monday. We agreed to meet on Mondays at 8:30 p.m. So inshallah we can solve these. أخيرا وليس آخرا، طبعا أنا هقابلكم تاني إن شاء الله في المحاضرة بتاعة الجينومكس يمكن في آخر الترم، ولعل وعسى إن إحنا هنتقابل يعني فيس تو فيس، الله أعلم. بس يعني كنت عايز أقول لكم إن إحنا وصلنا دلوقتي 4 محاضرات قابلناهم فيس تو فيس، أو ممكن تعدهم 8 بالكمبيوتر. طبعا هدفي زي ما أنا قلت لكم في الأول is not to give you enough information بس to make you curious to know more. وهدفي يعني is just to move the water. أنا بس يعني الكلام ده مش عارف أقوله باللغتين قوي، بس أنا كان كل هدفي زي ما اتفقنا في أول محاضرة إن أنا أحاول أحرك شوية بالنسبة لنا، أو shake the boat، أحرك المية كده بحيث إن إحنا نتعلم أكتر. فنفسي إن أنتوا never stop being curious، ما تبطلوش أبدا يبقى عندكم فضول. وإحنا دلوقتي أهو في مرحلة غريبة جدا من التاريخ. It's really useful for us to know microbiology, virology, and to know about immunology too. And remember بس إن الحياة مش linear. أنا قلت الكلام ده ومحضره من قبل حادثة الكورونا دي. فالحياة مش linear، بتبقى ماشي كده زي البكتيريا، وفجأة في lag phase وفجأة تبقى log phase. We never know what will happen to us tomorrow. So just don't stop having big dreams، وإن شاء الله بقية الكورس هتوفقوا فيه. فكده يعني أبقى أنا خلصت الجزء بتاع الميكروبيولوجي كله، وإن شاء الله نتقابل في أجزاء أخرى.